What is genetic engineering doing to our future? We are on the verge of rewriting the very code of life, curing diseases that have plagued us for millennia, creating crops that can feed a hungry planet, and maybe even unlocking a new era of human potential. Cut to darker, more ominous visuals, but this incredible, godlike power has a dark side. By tampering with the blueprint of nature, we risk unleashing new plagues, causing irreversible damage to our environment, and creating a world where humanity is split into the enhanced and the naturals. So, where is this technology actually taking us? The answer isn't simple, and it's both more hopeful and more worrying than you might think. The revolution is here. At the heart of every living thing is a manual, an instruction book written in a language billions of years old. This is DNA. It's the source code for life, with the precise instructions that build and run everything from a single bacterium to you and me. For most of human history, this code was an enigma locked away in our cells. We knew it dictated who we were and what diseases we might get, but we were powerless to change it. A single typo in that code could mean a life of suffering from a condition like cystic fibrosis or Huntington's disease, but that's not our reality anymore. The computer age gave us the tools to finally read this code, and in the last decade, we've developed a technology so revolutionary it has changed biology forever: CRISPR. Think of CRISPR as a find and replace function for our genes. It's like a pair of programmable molecular scissors that can scan through the billions of letters in an organism's DNA, find a specific sequence, and make a precise cut. This allows scientists to delete a harmful gene, repair a faulty one, or even insert a new one. The precision is stunning, and the implications are profound. This isn't science fiction, it's happening right now. The most celebrated success story is the fight against sickle cell disease, a painful, life-shortening blood disorder caused by one tiny error in the genetic code. Using CRISPR, scientists can take a patient's own blood stem cells, edit them in a lab to correct the faulty gene, and then return them to the patient. The results have been miraculous. The first ever approved CRISPR-based medicine, called Casgevy, is now being used to treat both sickle cell disease and another blood disorder, beta thalassemia. Clinical trials have shown that patients can be freed from the debilitating pain crises that once defined their lives. To go from the discovery of CRISPR to an approved therapy in just over a decade is a monumental achievement in medicine, and this is just the beginning. Clinical trials using CRISPR and other gene editing tools are underway to fight some of humanity's biggest foes. There are trials aimed at snipping the HIV virus out of a person's immune cells, though early results show this is an incredibly difficult challenge. Other researchers are engineering a patient's own immune cells into super-powered cancer hunters programmed to seek and destroy tumors. And there are promising therapies being tested for hereditary blindness, muscular dystrophy, and even high cholesterol. In a world first, a baby born in 2024 with a rare and fatal genetic disorder was treated with a personalized gene editing therapy developed in just 6 months to correct his specific mutation. We're witnessing the birth of truly personalized medicine, where treatments are designed not just for a disease, but for an individual's unique genetic makeup. The promise extends far beyond our own health. Genetic engineering is poised to tackle one of the biggest challenges of the 21st century, feeding a growing population on a warming planet. Scientists are developing crops that can thrive in drought, resist devastating infections, and produce higher yields. For example, genetically modified crops like Bt corn produce their own insecticide, which can dramatically reduce the need for chemical sprays. This has environmental benefits and helps protect farmers and beneficial insects from harmful pesticides. This is the golden age we were promised, a future where we can correct nature's deadliest mistakes and engineer a healthier, more sustainable world. But what if our corrections create even bigger mistakes? The dark side, this incredible power to rewrite life, comes with an equally incredible risk. The genome isn't a simple list of instructions. It's a deeply complex system that we're only just beginning to understand. Pulling on one thread can unravel another in ways we never intended. This is the dark side of genetic engineering, a future of unintended consequences we might be stumbling into. One of the first places we see these concerns is in our food. When you insert a new gene into a plant, you're telling it to produce a new protein. The question is, what else does that new protein do? In one infamous experiment, scientists transferred a gene from a Brazil nut into soybeans to make them more nutritious. The experiment worked, but with a terrifying side effect. People with nut allergies had life-threatening reactions to the soybeans because they now contained the Brazil nut allergen. And the very act of inserting a gene can disrupt a plant's stable code, potentially activating dormant genes that produce toxins. A case in the late 1980s serves as a stark reminder. A dietary supplement, L-tryptophan, was linked to an epidemic of a painful syndrome that led to dozens of deaths and thousands of permanent disabilities. While one of the manufacturers, Showa Denko, used genetically engineered bacteria in their new process, later investigations pointed to contaminants from inadequate purification as the likely cause, not the genetic engineering itself. Still, it shows how new production methods can introduce unexpected dangers. Beyond our plates, there's the risk of creating ecological problems. Many GM crops are engineered to resist herbicides like glyphosate. This lets farmers spray their entire fields, killing weeds but leaving the crop unharmed. The problem is, nature adapts. The widespread use of these crops has led to a massive increase in herbicide use, which in turn has sped up the evolution of superweeds that are also resistant to the spray. This forces farmers into a chemical arms race, using even more, and often harsher, chemicals to control them. Similarly, some pests are evolving resistance to the insecticides produced by crops like Bt corn, creating superpests that undermine the technology's original benefit. Perhaps the most haunting ecological risk is gene flow. Genes don't always stay put. Pollen from GM crops can drift for miles, fertilizing wild relatives or contaminating the fields of organic farmers. This can have real ecological costs. For instance, the widespread use of glyphosate on herbicide-resistant crops has been a major contributor to the decline of milkweed, the only food source for monarch butterfly caterpillars, which has severely impacted their population. An even more powerful and controversial technology called a gene drive could make these issues seem minor. A gene drive ensures a specific trait is passed down to nearly all offspring, allowing it to spread through a population with terrifying speed. Scientists have proposed using them to wipe out malaria-carrying mosquitoes or invasive species, but what if it goes wrong? A flawed or escaped gene drive could potentially alter or even eradicate an entire species, with unpredictable and irreversible consequences for the ecosystem. Because of this risk, all gene drive research is currently highly regulated and confined to labs. And this brings us to the most profound question of all, the designer-baby dilemma. If we can use CRISPR to correct a disease-causing gene, what stops us from using it to *enhance other traits, like height, intelligence, or athleticism? The technology to do this, called germline editing, already exists, and it makes changes that are heritable, passed down to all future generations. This creates the terrifying prospect of a new, biologically enforced class system. A world where the wealthy can afford to give their children a genetic head start, creating a society divided not by money, but by DNA. This ethical nightmare became a shocking reality in 2018 when a Chinese scientist, He Jiankui, announced he had created the world's first gene-edited babies, sparking global outrage and condemnation. He had crossed a line that many in the scientific community believed should never be crossed, opening a Pandora's box of ethical and societal risks. The crossroads, so this is where we stand at a monumental crossroads. In one direction is a future free from genetic disease, with abundant food and a healthier planet. In the other, a dystopian world of engineered plagues, ecological collapse, and a humanity fractured by a genetic caste system. The truth is, the technology itself is neutral. It's a tool, and the outcome depends entirely on the hands that wield it. The advance of this technology feels inevitable. The science is moving at a breathtaking pace, far outstripping our ability to create laws and regulations to govern it. The debate is no longer asterisk if asterisk we will use genetic engineering, but asterisk how asterisk. Will we establish clear ethical boundaries, agreed upon by the global community? Or will we allow a free-for-all, where a single person or corporation can make irreversible changes to the human gene pool or the global ecosystem? This isn't a choice for scientists and politicians alone. It's a choice for all of us. The decisions made in labs and regulatory bodies today will shape the future of our species and our planet for millennia. They will determine what it means to be human in the centuries to come. We are the first generation in the history of life on Earth with the power to consciously direct our own evolution. So the final question isn't just what is genetic engineering doing to our future? The real question is, what future will we choose to engineer? Will we use this power with wisdom, humility, and a profound sense of responsibility? Or will we, in our quest to build a perfect future, inadvertently engineer our own downfall? The code of life is now in our hands. The choice is ours.